Thank you. I'm excited to share some preliminary results from one of the first projects of my dissertation that explored drivers of phenotypic plasticity in Acropora cervicornis. Phenotypic plasticity, or the capacity of a single genotype to display a range of phenotypes, is important for corals' ability to respond to changing environmental conditions. And this capacity for different characteristics to result from the same genetic material is likely related to epigenetic mechanisms that perpetuate changes in gene activity in the context of the same DNA sequence. So far, the most studied epigenetic modification in corals is DNA methylation. This is a structural change where a methyl group is added to the DNA strand, usually at a cytosine base. DNA methylation plays a role in corals responding to environmental changes. It's related to the variability in gene expression and therefore the capacity for phenotypic plasticity and acclimatization. In this study, we explore how environmental and both epigenetic and genetic factors interact to influence the capacity for phenotypic plasticity. The study was completed on Bonaire and focused on 3 reef renewal nursery sites that varied in distance from the capital city, shown by the gray star. Bonaire experiences seasonal changes in temperature, as well as a rainy season in the fall and winter, and these predicted variations in environmental conditions were used as a natural experiment, exposing corals to both spatial and temporal differences in terrestrial runoff and water temperatures. At 4 seasonal time points, we monitored and sampled Acropora cervicornis corals while also documenting environmental conditions throughout the year. The 4 genotypes had been present at each site for years prior to the study, providing a unique system to assess genotype by environment interactions. To characterize coral phenotype, we performed an NMDS analysis of a range of coral physiological metrics. On the plot shown here, 1 point represents the physiology of one coral sample, and points that are closer together are more similar. Physiology varies significantly across coral genotype. Here, the density of points from each genotype is shown across the MDS1 axis. Coral physiology also varied across the 2 environmental factors of site and seasonal time point. We also found significant genotype by environment interactions, with genotypes responding differently to both temporal and spatial environmental variation, and this suggests that the capacity for plasticity may differ between genotypes. After finding that coral physiology was influenced by both genetic and environmental factors, we then compared those influences. And over 50% of the variance in coral physiology was explained by the 3 main variables, and a majority of that percentage was explained by the environmental factors. To characterize patterns of DNA methylation, we also performed an NMDS analysis, where each point now represents the DNA methylation profile of one coral sample. DNA methylation patterns differed significantly between each genotype and also varied across seasonal environmental conditions. We also again found significant genotype by environment interactions in DNA methylation profiles. And lastly, we compared how coral phenotype was related to both genetic and epigenetic factors, and found that the variation in coral physiology was significantly related to both coral genotype and DNA methylation. To summarize, coral phenotype was influenced by both genotype and the environment. And genotype by environment interactions are important to consider within restoration, as the performance of a particular genotype may differ between coral nursery or outplanting sites, and the response to the environment may differ between genotypes. DNA methylation patterns differed across seasonal time points, which supports the role of DNA methylation in seasonal acclimatization of Acropora cervicornis, which also is in line with some previous results from our lab that looked at Acropora cervicornis DNA methylation patterns in Puerto Rico. This study also adds to the growing body of evidence linking variation in coral physiology to variation in DNA methylation, suggesting the role of epigenetic mechanisms in coral phenotypic plasticity. I would like to thank all of the members of Dr. Aaron Lopez's Environmental Epigenetics Lab, our key collaborators, along with all of my funding sources for supporting this project. And thank you.
We have time for a question, I think, or two, if anyone has a question I'd like to ask Serena.
Yes.
How did you look at the methylation profile?
So this was with a method called MSAP, Methylation Sensitive Amplified Polymorphism. So it's not a sequencing method, but it uses two different primers that recognize the same sequence but have different sensitivities to methylation. And so you get more of like the profile. You know that this one has methylation at that point and this one does not, but we didn't actually sequence where those were in the genome. So this is a method that you can process more samples, kind of with larger sample size, larger studies, but without as much of that like gene-specific information.
Great, thank you. I'm going to move us along for our next speaker. Please big thanks, Serena, once again. Thank you. Next, we have Martin.